Hello guys, welcome to my YouTube channel. So today we are going to learn some of the basics of Biopython. So this is the first series of Biopython lectures. So if you know the basics of Python, so it will be easier for you. Here we will analyze biological sequences with Python. So that's why it's called as Biopython. So since this is the first lecture, we will start with the very basic things and then gradually as the lecture series will progress, we will move towards the more complicated biopython functions. So first, this is a Google Colab, so even if you have Jupyter notebook, it's completely fine. So first, to work with biopython, we have to install the package, okay, of biopython. So we will write this import pip install biopython, okay. So if you're working with Google Colab, after writing your command, you have to press Shift Enter to run this cell. So as you can see here, this is running. Okay, so this cell is running. This cell is getting executed. So you wait until this cell gets executed and like all the necessary packages get installed. Only then you can use the functions and modules installed in those packages for analyzing your sequences. So here as you can see here, they are showing collecting biopython, downloading biopython this version, which consumes a memory of 3.1 MB. Okay, so yeah, so installing collected packages biopython, and it's showing successfully installed biopython. So biopython package downloading is done, and on the left hand side of the cell you will see a green tick, and it also shows the time taken to execute this cell, which is 15 seconds. So green tick signifies that your this cell has like execution has been completed. So you have installed all your packages. Okay, so now from this package we will load one of our packages. Okay, so I'm writing the comment here, load our bioinformatics package. Okay, so we will write it as import bio and then shift enter. So this is the package that we are importing from this module biopython. Okay, so then we will write some methods and attributes. So these greenish ones, which I am writing by giving a hash sign. So these are comments that I'm putting for you to understand. So dir, so the command is dir bio. Okay, and then shift enter. So now this cell is running. So you can see these are the operations we can perform in biopython. So sorry, these are the attributes. So these are the attributes that are there in the biopython. Okay, so now we will now see how to work with a particular sequence in biopython. Okay, so what we'll do, we will convert this code cell into a text cell just for your understanding. So we will write it here as working with sequences. Okay, so this is done. So from here I put heading. So from here whatever we write the code, that will be the code of how to work with sequence. Okay, so from the bio module, from the bio package, bio module, we import one package which is sequence IO. So we'll write the command as from bio import sequence IO, so seqio. Okay, and we will run this cell, shift enter. So it's running. So done. And from this So the next line will be from bio dot sequence. So we are actually accessing by the using the dot operator, we are accessing this bio dot sequence. So we are accessing this sequence package in bio. Okay, and we are again importing another package which is import sequence. Okay, so this we are running, and now we will see what are the attributes, what can be the attributes of sequence package. So next line will be methods of sequence. Okay, dir sequence. So these are the attributes we can get from a particular sequence. If you have a sequence, we can get that item, we can know which subclass it belongs, which module it belongs, we can know the size of that particular sequence, like that. So these are the possible attributes that we can get. Now next we will, okay, so we will create a particular or create a simple sequence. So I'm writing here that create a simple sequence. Okay, I'm storing it in a temporary variable called a sequence1, and I'm accessing the package which is seq, and within brackets I'm giving my sequence. So suppose I'm creating a sequence called a t g a t c t, okay, c g t a, maybe this one sequence which I am creating, and it will get stored in this temporary variable sequence1. I'm running it. This cell is executed. Suppose if I want to see whether this sequence has really been stored in this temporary variable sequence1, what will I do? I'll just write sequence1 and run this code cell. So let's, yeah, so it's stored. So now I will create a generic DNA similarly that I did here. So I'll write here as create a generic DNA. Okay, and here I'll write from bio.sequence. It's fine, we have written it earlier also, but it's fine if you don't write it again, but I'm writing just for the sake in case you missed it. Okay, and then I am giving the DNA as sequence same. Okay, so this sequence only we can use. This was an example, but I'm now doing for a DNA, so I'm writing it again. Okay, and we are running it. Done, code executed. We'll check if it's really in store or not. Okay, so yeah, it's done. So now we can similarly we can create one generic RNA also. Okay, so what will we do? Instead of this thymine, we will just put uracil. So I'm just copying it here. This command, I'm copying whatever is in this cell. I'll just change everything to RNA. So instead of DNA, it will be RNA, and this will be RNA, and instead of all the T's, you will have to put U's. Okay, so yeah, there are 4 T's, so there will be 4 U's, and I'm running this cell. So done. Let's check once. Done. Okay, so RNA sequence we have, DNA sequence we have, so this is how we can create. Now suppose we are intending to find the length of our DNA sequence. So how will you find? So this is the code of length of DNA sequence. This is very simple. You will have to write the command len. Len stands for finding the length. Within brackets you give the variable name and you run this cell. Okay, so 12. So this is the length of the DNA sequence is 12. So if you notice here, there are all total I guess there are 12 nucleotides, so that's why it's shown the length is 12. Okay, now if suppose you're interested for slicing. Okay, slicing, if you do, so this is the code for the slicing. You have the DNA and that sequence that you got, that you have, you want to slice it. So you will write DNA within third brackets 0:3. Okay, and you press shift enter. Let's see what happens. So sequence ATG. Okay, so you slice but some particular portion. So what did you do? 0 means, so this is basically some sort of a list. Okay, so this was your DNA sequence. So you slice the first 3 nucleotides out of the 12 nucleotides. So the list starts from actually it's a tuple, so it starts from 0th position. So this A is the 0th position, then T is at the first position, G is the second position. Okay, so here you are writing DNA 0 up to 3. Okay, but not 3. So it will show the output from 0 to 2. Okay, 0:3 means the numbering is first position is 0th, then first, the second position is designated as first, third position designated as second. Okay, and fourth position designated as third. So 0:3 means it will not show the fourth position, but it will show till the third position. So here 0th, first and second, so first 3 will be shown. Okay, so the indexing starts from 0, and you will 0:3 means 0 indexing, first indexing, second indexing only. So from 0th till third, but not third indexing it will show. So suppose you are interested in reversing. Okay, reversing the sequence. So what will you do? You will just write DNA. Okay, that sequence that you have. Okay, you will put 2 colons and write -1. So -1 means everything gets reversed. You run this code. So see, if you know this, this was your original sequence. I will go again up. So this was your original sequence, this was your sequence. Okay, and now here you have reversed everything. So this is your reverse sequence. So the adenines which were at the last, now they are at the first positions. Okay, so this is how you can reverse your sequence. So remember, for one example, before moving on to the DNA sequence, I showed you this sequence 1 example. So this was just an example, and later I used this sequence only for creating the DNA sequence. So these two basically the same, but we also have this sequence stored in another variable called sequence 1. Okay, so you remember we have one sequence 1 also. Okay, so this sequence is basically stored in 2 locations. One is sequence 1 and one is the DNA variable. So because we have sequence 1 also, so I will create another sequence called sequence 2. Okay, why I am creating it, I will show you. So sequence 2. Okay, so I'm giving another sequence A T G A T C T C G T G G. Okay, and I'm running this cell. So done, execution complete. We'll check once. Okay, stored. Now we have two sequences, sequence 1 and sequence 2. Now suppose you intend to join the two sequences. So this code shows you the joinings of the two sequences. Okay, how will you join? So you will write DNA, or maybe the sequence 1. Let's not get confused. So sequence 1. Okay, and within third brackets you write 0 till 6 plus sequence 2. So the indexing, it will take the nucleotides from 0th indexing till 5th indexing. 0:6 means 1 before. So 0th, 1st, 2nd, 3rd, 4th, 5th. So 5th indexing it will take, and then it will amalgamate to the entire sequence 2. Okay, we'll run this. Yeah, so see, this is the combined sequence after combining the two sequences. So what we have done is we have combined some nucleotides from the first sequence with all the nucleotides from the second sequence. Okay, so you could have, what you could have done is you could have just joined the entire nucleotides. So in that case, what would have you done? You will just write sequence 1 plus sequence 2. Just run. Yeah. So this is the entire sequence. So we have not omitted anything. Entire sequence 1 combined with sequence 2. So there are 24 nucleotides. Okay. Okay. Now suppose we intend to count how many nucleotides are there. So suppose we intend to count how many adenine nucleotides are there in the DNA sequence, or how many guanine nucleotides are there in the DNA sequence. So you remember, I will go again up. So this was what our DNA sequence was. So this is our DNA sequence. So I want to know how many adenines are there, how many guanines are there, how many thymines are there. Okay. So what will we do? Just a second. So this find, okay, so find the number of nucleotides within our sequence. Okay, so yeah, so how will we do? We will use, so we'll actually basically we are counting. So once again, for your convenience, I'm writing the DNA sequence. Okay, so what was our DNA sequence? So this is what is stored in the DNA variable. Okay, now I want to count how many guanines are there. So I will write DNA dot count. So as you know, this dot operator is used to access the individual members of this tuple. So DNA dot count, and I'll write within quotations G, and I'll run it. So it shows two. So see, there are two guanines within this sequence, this and this. Okay, similarly, if I'm interested to count how many adenines are there. So I'll write adenines and run this cell. So it shows 4. Okay, so I can even count how many uh, number of codons are there within a sequence. Okay, same thing, we'll use the count. So DNA dot count, and here instead of putting one, just one member, we will put 3 together. So 3 nucleotides, and run this. So it will search for ATG as a whole. So we have ATG as a whole in only like one place. So the output is 1. Okay. So likewise, we'll proceed with the others. So suppose we are instead of counting how many guanines there, we intend to find out what is the position of guanine. Okay, so how will we write it? So let's see. uh, so find the position of a nucleotide or a location. So we will use the find, DNA dot find. Okay, guanine. So the guanine is present at 2 positions. Okay, so here as you can see, one position is here and another position is here. Okay, so it shows that guanine is present at 2 positions. So suppose we intend to find the position or location of a nucleotide from the right. So we will write it as this thing only, but we'll write from right. So we will write DNA dot, yeah, so it's written already, DNA dot r find, and we'll run this cell. Let's see what happens. So it's showing E. If you are intending to find the location or exact position or the index of a particular say nucleotide, so find the index of the nucleotide. Okay, so what will we do? We will write DNA dot index. So we'll run this cell, so it will show the index, which is 2. Remember that list of tuples starts from 0th. Okay, so 2 means third position. Okay, so similarly we can find the location or the index from the right also. So here we will write from the right, and this here we will just put r index. Okay, dot r index, and we'll run this cell. Okay, now one last thing, suppose we want to get a bar graph of the variation of nucleotides. So let's see, plot the frequency of nucleotides. Okay, so for plotting we have to use matplotlib. So import matplotlib.pyplot as plt. Okay, so we'll run this. So this is basically used for plotting in Python. So again we have to write from collections, import counter. So we have to count individual nucleotides, that's why you're importing counter. Then this was our DNA sequence. If you remember it, I'm writing it again. Then I'll write counter DNA. Counter, sorry, C in capital, counter DNA. I'll just run it. So what will happen, it will like count every nucleotide. So there are 4 A's, 4 T's, 2 G's, 2 C. Okay, so with this we can like find out how many adenines, guanines, thymine, cytosine are there in a single step. So we don't need to again we write 4 different codes for finding the adenine, guanine that is showed earlier. So in a single step we can do it. Now we will find the frequency. So we'll write DNA underscore frequency is equal to counter DNA. Okay, so whatever the counts of each individual nucleotide, we are storing it in a new variable called as the DNA frequency. We are running it here. Okay, and let's see if it gets stored. So we are running this cell. Let's see, same thing. So the values, okay, so this entire thing has been stored in a new variable DNA frequency. Now we'll plot. So for plotting we'll write as a bar graph we are plotting, so we'll write plt.bar. So we will plot these frequencies or the counts. Okay, frequencies dot keys comma DNA frequency dot values. Okay, so DNA underscore frequency dot keys means this A, T, G, C. So it will label your graph. So you will understand which bar graph corresponds to which nucleotide. and DNA underscore frequency dot values will take the values this 4422 these things, and we'll press shift enter. So here this is the bar graphs. As you can see here, you have 4 adenines and 4 thymines. So these two are the highest bar graphs, and they both are equal and corresponds to this 4 value. Okay, whereas you have only 2G and 2Cs, so these two are equal because each one of them we have 2 and this bar graph corresponds to 2 values. So if you have a particular sequence, or suppose you are intending to like combine two sequences, so you can find out the frequency in this like this plot. Okay, so you can also like plot two different sequences simultaneously side by side and compare them. Like if you have another sequence, you can plot it in the same graph cluster as you can do in Excel, and then you can compare, okay, this sequence has high saturity, this sequence is having lower guanine, and like that. Okay, so thank you for attending this is the first session of Biopython. We will see other problems, we'll solve other problems in subsequent lectures also.